Chapter 16 Chemistry in Everyday Life. By now, you have learned the basic principle of chemistry and also realized that it influences every sphere of human life. The principles of chemistry have been used for the benefit of mankind. Think of cleanliness, the materials like soaps, detergents, household bleaches, toothpaste, etc. will come to your mind. Look toward the beautiful clothes immediately. Chemicals of the synthetic fibers used for making clothes and chemicals giving colors to them will come to your mind. Food materials, again, a number of chemicals about which you have learned in the previous unit will appear in your mind. Of course, sickness and diseases remind us of medicine. Again, chemicals. Explosives, fuels, rocket propellants, building and electronic materials, etc. are all chemicals. Chemistry has influenced our life so much that we do not even realize that we come across chemicals at every moment. That we ourselves are beautiful chemical creations and all our activities are controlled by chemicals. In this unit, we shall learn the applications of chemistry in three important and interesting areas, namely medicines, food materials, and cleansing agent. 16.1 Drugs and their classifications. Drugs are chemicals of low molecular masses. These interact with macromolecular targets and produce a biological response. When the biological response is therapeutic and useful, these chemicals are called medicines and are used in diagnosis, prevention, and treatment of diseases. If taken in doses higher than those recommended, most of the drugs used as medicines are potential poisons. Use of chemicals or therapeutic effect is called chemotherapy. 16.1.1 Classification of drugs. Drugs can be classified mainly on criteria outlined as follows. A. On the basis of pharmacological effect. This classification is based on pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for the treatment of particular type of problem. For example, analgesics have pain-killing effect, antiseptic kill or arrest the growth of microorganisms. B. On the basis of drug action. It is based on the action of drug on a particular biochemical process. For example, all antihistamines inhibit the action of compound histamine which causes inflammation in the body. There are various ways in which action of histamines can be blocked. You will learn about this in section 16.3.2. C. On the basis of chemical structure. It is based on the chemical structure of the drug. Drugs classified in this way share common structural features and often have similar pharmacological activity. For example, sulfonamides have common structural feature given below. A diagram. D. On the basis of molecular targets. Drugs usually interact with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. These are called target molecules or drug targets. Drugs possessing some common structural features may have the same mechanism of action on drugs. The classification based on molecular targets is the most useful classification for medicinal chemists. 16.2 Drug target interaction. Macromolecules of biological origin perform various functions in the body. For example, proteins which perform the role of biological catalysts in the body are called enzymes. Those which are crucial to communication system in the body are called receptors. Carrier proteins carry polar molecules across the cell membrane. Nucleic acids have coded genetic information for the cell. Lipids and carbohydrates are a structural part of the cell membrane. We shall explain the drug target interaction with the example of enzymes and receptors. Right. 16.2.1 Enzymes as drug target. A. Catalytic action of enzymes. For understanding the interaction between a drug and an enzyme, it is important to know how do enzymes catalyze the reaction. In their catalytic activity, enzymes perform two major functions. First, the first function of an enzyme is to hold the substrate for a chemical reaction. Active sites of enzymes hold the substrate molecule in a suitable position so that it can be attacked by the reagent effectively. Substrates bind to the active site of the enzyme through a variety of interactions such as ionic bonding, hydrogen bonding, van der Waals attractions, or dipole-dipole interactions. The second function of an enzyme is to provide functional groups that will attack the substrate and carry out chemical reactions. B. Drug enzyme interaction. Drug inhibits any of the above mentioned activities of enzymes. These can block binding sites of the enzyme and prevent the binding of substrate or can inhibit the catalytic activity of the enzyme. Such drugs are called enzyme inhibitors. Drugs inhibit the attachment of substrate or active site of enzymes in two different ways. First, drugs compete with the natural substrate of their attachment on the active sites of enzymes. Such drugs are called competitive inhibitors. Second, some drugs do not bind to the enzyme's active site. These bind to a different site of enzyme which is called allosteric site. This binding of inhibitor at allosteric site changes the shape of active site in such a way that substrate cannot recognize it. If the bond formed between an enzyme and an inhibitor is strong, covalent and very much strong, cannot be broken easily, then enzyme is blocked permanently. The body then degrades the enzyme inhibitor complex and synthesizes the new enzyme. 16.2.2 Receptors as drug targets. Receptors are proteins that are crucial to body's communication process. Majority of these are embedded in cell membranes. Receptor proteins are embedded in the cell membrane in such a way that their small part possessing active site projects out of the surface of the membrane and opens on the outside region of the cell membrane. In the body, message between two neurons and that between neurons to muscles is communicated through certain chemicals. These chemicals, known as chemical messengers, are received at the binding sites of receptor proteins. To accommodate a messenger, the shape of the receptor site changes. This brings about the transfer of message into the cell. Thus, chemical messengers gives message to the cell without entering into the cell. There are a large of number of different receptors in the body that interact with the different chemical messengers. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure and amino acid compositions. Drugs that bind to the receptor site and inhibit its natural function are called antagonists. These are useful when blocking of message is required. There are other type of drugs that mimic the natural messenger by switching on the receptor. These are called agonists. These are useful when there is a lack of natural chemical messengers. 16.3 Therapeutic actions of different classes of drugs. In this section, we will discuss the therapeutic action of a few important classes of drugs. 16.3.1 Antacids. Overproduction of acid in the stomach causes irritation and pain. In severe cases, ulcers are developed in the stomach. Until 1970, only treatment for acidity was administration of antacids such as sodium hydrogen carbonate or a mixture of aluminum and magnesium hydroxide. However, excessive hydrogen carbonate can make the stomach alkaline and trigger the production of even more acid. Metal hydroxides are better alternatives because of being insoluble. These do not increase the pH above neutrality. These treatments control only symptoms and not cause. Therefore, these metal salts, the patients cannot be treated easily. In advanced stages, the ulcers become life-threatening and its only treatment is removal of the affected part of the stomach. A major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical histamine stimulates the secretion of pepsin and hydrochloric acid in the stomach. The drug cimetidine, also called as Tigamet, was designed to prevent the interaction of histamine with the receptor present in the stomach wall. This resulted in release of lesser amount of acid. The importance of the drug was so much that it remained the largest selling drug in the world until another drug, ranitidine, also called as Zemtec, was discovered. 16.3.2 Antihistamines. Histamine is a potent vasodilator. It has various functions. It contracts the smooth muscles in the bronchi and gut and relaxes other muscles, such as those in the walls of fine blood vessels. Histamine is also responsible for the nasal congestion associated with common cold and allergic response to pollen. Synthetic drugs brompheniramine, also called as Dimetapp, and terfenadine, also called as Seldane, act as antihistamines. They interact with the natural action of histamine by competing with the histamine for binding sites of receptor where histamine exerts its effect. Now the question that arises is, why do above mentioned antihistamine not affect the secretion of acid in the stomach? The reason is that anti-allergic and antacid drugs work on different receptors. 16.3.3 Neurological active drugs. A. Tranquilizers. Tranquilizers and analgesics are neurological active drugs. These affect the message transfer mechanism from nerve to receptor. Tranquilizers are a class of chemical compound used for the treatment of stress and mild or even severe mental diseases. These relieve anxiety, stress, irritability, or excitement by including or inducing a sense of well being. They form an essential component of sleeping pills. These are various types of tranquilizers. They function by different mechanisms. For example, noradrenaline is one of the neurotransmitters that plays a role in mood changes. If the level of noradrenaline is low for some reason, then the signal sending activity becomes low and the person suffers from depression. In such situations, antidepressant drugs are required. These drugs inhibit enzymes which catalyze the degradation of noradrenaline. If the enzyme is inhibited, this important neurotransmitter is slowly metabolized and can activate its receptor for longer periods of time, thus counteracting the effect of depression. Iprionazid and phenelazine are two such drugs. Some tranquilizers, namely chlordiazepoxide and meprobamate, are relatively mild tranquilizers suitable for relieving tension. Equinil is used in controlling depression and hypertension. Derivatives of barbituric acid like veronal, amityl, nibutyl, nummulyl, and seconal constitute an important class of tranquilizers. These derivatives are called barbiturates. Barbiturates are hypnotic, that is, sleep-producing agents. Some other substances used as tranquilizers are Valium and serotonin. B. Analgesics. Analgesics reduce or abolish pain without causing impairment of consciousness, mental confusion, incoordination, or paralysis, or some other disturbances of nervous system. These are classified as follows. First, non-narcotic analgesics. Second, narcotic drugs. First, non-narcotic analgesic. Aspirin and paracetamol belong to the class of non-narcotic analgesics. Aspirin is the most familiar example. Aspirin inhibits the synthesis of chemicals known as prostaglandins, which stimulate inflammation in the tissue and cause pain. These drugs are effective in relieving skeletal pain such as that due to arthritis. These drugs in relieving skeletal pain such as these drugs have many other effects such as reducing fever, antipyretic, and preventing platelet coagulation. Because of its anti-blood clotting action, aspirin find use in prevention of heart attacks. Second, narcotic analgesics. Morphine and many of its homologs, when administered in medicinal doses, relieve pain and produce sleep. In poisonous doses, these produce stupor, coma, convulsions, and ultimately death. Morphine narcotics are sometimes referred to as opiates since they are obtained from the opium poppy. These analgesics are chiefly used for the relief of postoperative pain, cardiac pain, and pains of terminal cancer and in childbirth. 16.3.4 Antimicrobials. Disease in human beings and animals may be caused by a variety of microorganisms such as bacteria, virus, fungi, and other pathogens. An antimicrobial tends to destroy, prevent the development of, inhibit the pathogenic action of microbes such as bacteria, antibacterial drugs, fungi, virus, or other parasites selectively. Antibiotics, antiseptic, and disinfectant are antimicrobial drugs. A. Antibiotics. Antibiotics are used as such drugs to treat infections because of their low toxicity for humans and animals. Initially, antibiotics were classified as chemical substances produced by microorganisms like bacteria, fungi, and molds that inhibit the growth or even destroy microorganisms. The development of synthetic methods has helped in synthesizing some of the compounds that were originally discovered as products of microorganisms. Also, some purely synthetic compounds have antibacterial activity and therefore definition of antibiotic have been modified. An antibiotic now refers to a substance produced wholly or partly by chemical synthesis, which in low concentration inhibits the growth or destroy microorganisms by intervening in their metabolic processes. The search for chemicals that would adversely affect invading bacteria but not the host began in the 19th century. Paul Ehrlich, a German bacteriologist, conceived this idea. He investigated arsenic based structure in order to produce less toxic substance for the treatment of syphilis. He developed this medicine, arsphenamine, or known as salvarsan. Paul Ehrlich got Nobel Prize for Medicine in 1908 for the discovery. It was the first effective treatment discovered for syphilis. Although salvarsan is toxic to human beings, its effect on the bacteria Spirochete, which causes syphilis, is much greater on human beings. At the same time, Elric was working on agiodice also. He noted that there is similarity structure of salvarsan and agiodice. The As double bond As linkage present in the arfesinamine resembles that nitrogen double bond nitrogen linkage present in the aziodize in the sense that arsenic atom is present in place of nitrogen. He also noticed tissue getting colored by dyes selectively, therefore Ilric began to search for the compound which resemble in the structure to aziodize and selectively bind to bacteria. In 1932, he succeeded in preparing the first effective antibacterial agent, protonsil, which resembled in the structure to the compound salvarsan. Soon it was discovered that in the body protonsil is converted to a compound called sulfanilamide, which is the real active compound. Thus, the sulfa drugs were discovered. A large range of sulfonamide analogs was synthesized. One of the most effective is sulfapyridine. Despite the success of sulfonamides, the real revolution in antibacterial therapy began with the discovery of Alexander Fleming in 1929 of the antibacterial properties of penicillium fungus. Isolation of purification of active compound to accumulate sufficient material for the clinical trials took 13 years. Antibiotics have either cidal killing effect or static inhibitory effect on microbes. A few examples of the types of antibiotics are as follows. First one is bactericidal, which contains three penicillin aminoglycosides of fluoxetine. And second one is bacteriastatic, that contains three erythromycin, tetracycline, and chloramphenicol. The range of bacteria or other microorganisms that are affected by certain antibiotic is expressed as its spectrum of action. Antibiotics which kill or inhibit a wide range of gram-positive or gram-negative bacteria are said to be broad-spectrum antibiotics. Those effective mainly against gram-positive or gram-negative bacteria are narrow-spectrum antibiotics. If effective against a single organism or disease, they are referred to as limited-spectrum antibiotics. Penicillin G has a narrow spectrum. Ampicillin and amoxicillin are synthetic modification of penicillins. These have broad spectrum. It is absolutely essential to test the patients for sensitivity or allergy to penicillin before it is administered. Chloramphenicol, isolated in 1947, is a broad-spectrum antibiotic. It is rapidly absorbed from the gastrointestinal tract, hence can be given orally in case of typhoid, dysentery, acute fever, certain form of urinary infections, and pneumonia. Vancomycin and ofloxacin are other important broad-spectrum antibiotics. The antibiotic disadezirin is supposed to be toxic towards certain strains of cancer cell. B. Antiseptics and disinfectants. Antiseptic and disinfectants are also the chemicals which either kill or prevent the growth of microorganisms. Antiseptics are applied to the living tissues such as wound, cuts, ulcers, and diseased skin surfaces. Examples are furosemide, sulfamycin, etc. These are not ingested like antibiotics. Commonly used for the antiseptic Dettol is a mixture of chloroxylenol and terpinol. Bithenol, the compound is also called bithionol, is added to soaps to impart antiseptic properties. Iodine is a powerful antiseptic. Its 2-3% solution in a alcohol water mixture is known as tincture of iodine. It is applied on wounds. Hydroform is also used as an antiseptic for wounds. Boric acid in dilute aqueous solution is weak antiseptic for eyes. Disinfectant are applied to inanimate objects such as floors, drainage system, instrument, etc. Same substances can act as an antiseptic as well as disinfectant by varying the concentration. For example, 0.2% solution of phenol is an antiseptic while its 1% of solution is disinfectant. Chlorine in the concentration of 0.2 to 0.4 ppm in aqueous solution and sulfur dioxide in very low concentration are disinfectant. 16.3.5 Anti-fertility drugs. Antibiotic revolution has provided long and healthy life to people. The life expectancy has almost doubled. The increased population has caused many social problems in terms of food resources, environmental issues, employment, etc. To control these problems, population is required to be controlled. This has led to the concept of family planning. Anti-fertility drugs are of use in the direction. Birth control pills essentially contain a mixture of synthetic estrogen and progesterone derivatives. Both of these compounds are hormones. It is known that progesterone suppresses ovulation. Synthetic progesterone derivatives are more potent than progesterone. Norethindrone is an example of synthetic progesterone derivative most widely used as anti-fertility drug. The estrogen derivative which is used in combination with progesterone derivative is ethinyl estradiol or also called as noestrol. 16.4 Chemicals in food. Chemicals are added to food for their preservation, enhancing their appeal, adding nutritive value in them. Main category of food additive are as follows. First, food colors. Second, flavors and sweeteners. Third, fat emulsifiers and stabilizing agent. Fourth, flour improvers, anti-stalling agents and bleaches. Fifth, antioxidants. Sixth, preservatives. And seven, nutritional supplements such as minerals, vitamins, and amino acids. Except for chemical of category 7, none of the above additives have nutritive value. These are added either to increase the shelf life of stored food for cosmetic process or purposes. In this section, we will discuss only sweeteners and food preservatives. 16.4.1 Artificial sweetening agent. Natural sweetness example sucrose add to calorie intake and therefore many people prefer to use artificial sweeteners. Orthosulfonbenzoamide, also called as saccharine, is the first popular artificial sweetening agent. It has been used as a sweetening agent ever since it was produced or discovered in 1870. It was about 550 times as sweet as cane sugar. It is excreted from the body in urine unchanged. It appears to be entirely inert and harmless when take. Its use is of great value of diabetic person and people who need to control intake of calories. Some other commonly marketed artificial sweetening agents are given in table. These are aspartame, saccharine, sucralose, and alitium. Aspartame is the most successful and widely used artificial sweetener. It is roughly 1000 times or 100 times as sweet as cane sugar. It is methyl ester of dipeptide formed from aspartic acid and phenylalanine. Use of aspartame is limited to cold foods and soft drinks because it is unstable at cooking temperature. Alitium is high potency sweetener. Although it is more stable than aspartame, the control of sweetness of food is difficult while using it. Sucralose is trichloro derivative of sucrose. Its appearance and taste are like sugar. It is stable at cooking temperature. It does not provide calories. 16.4.2 Food preservatives. Food preservative prevent spoilage of food due to microbial growth. The most commonly used preservative include table salt, sugar, vegetable oils, sodium benzoate. Sodium benzoate is used in limited quantities and is metabolized in the body. Salts, ascorbic acid, and propanoic acid are also used as preservatives. 16.5 Cleansing agents. In this section, we will learn about detergents. Two types of detergents are used as cleansing agent. These are soap and synthetic detergents. The improved cleansing agent of water. These help in removal of fat which binds other materials to the fabric or skin. Soaps are detergents used since long. Soaps used for cleansing purpose or sodium or potassium salts of long chain fatty acid. Example: stearic, oleic, palmitic acid. Soaps containing sodium salts are formed by heating with aqueous sodium hydroxide solution. This reaction is known as saponification. In this reaction, ester of fatty acid are hydrolyzed and soap obtained remains in colloidal form. It is precipitated from the solution by adding sodium chloride. The solution left after removal the soap contains glycerol, which can be recovered by fractional distillation. Only sodium and potassium soaps are soluble in water and are used for cleaning purposes. Generally, potassium soaps are soft to the skin than sodium soaps. These can be prepared by using potassium hydroxide solution in place of sodium hydroxides. Types of soaps. Basically, all soaps are made by boiling fats or oils with suitable soluble hydroxide. Variations are made by using different raw materials. Toilet soaps are prepared by using better grades of fats and oils, and care is taken to remove excess alkali. Color and perfumes are added to make these more attractive. Soaps that float in water are made by beating tiny air bubbles before their hardening. Transparent soaps are made by dissolving the soap in the ethanol and then evaporating the excess solvent. In medicated soaps, the substances of medicinal value are added. In some soaps, deodorants are added. Shaving soaps contain glycerol to prevent rapid drying. A gum called rosin is added while making them. It forms sodium rosinate, which lather well. Laundry soap contains filler like sodium rosinate, sodium silicate, borax, and sodium carbonate. Soap chips are made by running a thin sheet of melted soap onto a cool cylinder and scraping off the soaps in small broken pieces. Soap granules are dried miniature soap bubbles. Soap powder and scoring soaps contain some soap, a scoring agent such as powdered pumice or finely divided sand, and builders like sodium carbonate and trisodium phosphate. Why do soaps do not work in hard water? Hard water contains calcium and magnesium ions. These ions are insoluble calcium and magnesium soaps respectively when sodium or potassium soaps are dissolved in hard water. These insoluble soaps separate as scum in water and are useless as cleansing agent. In fact, these are hindrance to good washing because the participate adheres onto the fiber of the cloth as gummy mass. Hair washed with the hard water looks dull because of this sticky precipitation. Dye does not absorb evenly on cloth washed with soap using hard water because of this gummy mass. 16.5.2 Synthetic detergents. Synthetic detergents are cleansing agents which have all the properties of soap but which actually do not contain any soap. These can be used both in soft and hard water as they give foam even in hard water. Some of the detergents give foam in ice cold water. Synthetic detergents are mainly classified into three categories. First, anionic detergent, second, cationic detergent, and third, non-ionic detergents. First, anionic detergents. Anionic detergents are sodium salts of sulfonated long-chain alcohols or hydrocarbons. Alkyl hydrogen sulfates formed by treating long-chain alcohols with concentrated sulfuric acid are neutralized with alkali to form anionic detergents. Similarly, alkyl benzene sulfonates are obtained by neutralizing alkyl benzene sulfonic acid with alkali. In anionic detergents, the anionic detergent of the molecule is involved in the cleansing action. Sodium salts of alkene benzene sulfonates are important class of anionic detergent. They are mostly used for household work. Anionic detergents are also used in toothpaste. Second, cationic detergents. Cationic detergents are quaternary ammonium salts of amines with acetates, chlorides or bromides as anions. Cationic part possesses a long hydrocarbon chain and a positive charge on the nitrogen atom. Hence, these are called cationic detergents. Cetyl methyl ammonium bromide is a popular cationic detergent and is used in hair conditioner. Cationic detergents have germicidal properties and are expensive, therefore, these are of limited use. Third, non-ionic detergents. Non-ionic detergents do not contain any ion in their constitution. One such detergent is formed when stearic acid reacts with polyethyl glycol. Liquid dishwashing detergents are non-ionic type. Mechanism of cleansing action of this type of detergent is of the same as that of soaps. These also remove grease and oil by micelle formation. Main problem that appears in the use of detergent is that if their hydrocarbon chain is highly branched, then bacteria cannot degrade. Slow degradation of detergents lead to their accumulation. Effluents containing such as detergents reach the river, ponds, etc. These persist in water even after sewage treatment and cause foaming in rivers, ponds and streams, and their water gets polluted. These days, and the branching of the hydrogen carbon chain is controlled and kept in the minimum. Unbranched chain can be biodegraded more easily and hence polluted is prevented. Thank you.